clinical trials in China to treat head and neck cancers. The results have been promising, with a greater short-term response rate to the combination of chemotherapy and viral therapy than to chemotherapy treatment alone. This ongoing research may herald the beginning of a new age of cancer therapy, where viruses are engineered to find and specifically kill cancer cells, regardless of where in the body they may have spread. A third use of viruses in medicine relies on their specificity and involves using bacteriophages in the treatment of bacterial infections. Bacterial diseases have been treated with antibiotics since the 1940s. However, over time, many bacteria have evolved resistance to antibiotics. A good example is methicillin-resistant Staphylococcus aureus, MRSA, pronounced MRSA, an infection commonly acquired in hospitals. This bacterium is resistant to a variety of antibiotics, making it difficult to treat. The use of bacteriophages specific for such bacteria would bypass their resistance to antibiotics and specifically kill them. Although phage therapy is in use in the Republic of Georgia to treat antibiotic-resistant bacteria, its use to treat human diseases has not been approved in most countries. However, the safety of the treatment was confirmed in the United States when the U.S. Food and Drug Administration approved spraying meats with bacteriophages to destroy the food pathogen Listeria. As more and more antibiotic-resistant strains of bacteria evolve, the use of bacteriophages might be a potential solution to the problem, and the development of phage therapy is of much interest to researchers worldwide. 526 Chapter 21, Viruses Access for Free at OpenStax.org. Loss of motor control and unusual behaviors, such as uncontrolled bursts of laughter with Kuru, followed by death. Kuru was controlled by inducing the population to abandon its ritualistic cannibalism. On the other hand, BSE was initially thought to only affect cattle. Cattle dying of the disease were shown to have developed lesions or holes in the brain, causing the brain tissue to resemble a sponge. Later on in the outbreak, however, it was shown that a similar encephalopathy in humans, known as variant Creutzfeldt-Jakob disease, CJD, could be acquired from eating beef from animals infected with BSE, sparking bans by various countries on the importation of British beef and causing considerable economic damage to the British beef industry. Figure 21.17. BSE still exists in various areas, and although a rare disease, individuals that acquire CJD are difficult to treat. The disease can be spread from human to human by blood, so many countries have banned blood donation from regions associated with BSE. The cause of spongiform encephalopathies, such as Kuru and BSE, is an infectious structural variant of a normal cellular protein called PRP, prion protein. It is this variant that constitutes the prion particle. PRP exists in two forms, PRPC, the normal form of the protein, and PRPSC, the infectious form. Once introduced into the body, the PRPSC contained within the prion binds to PRPC and converts it to PRPSC. This leads to an exponential increase of the PRPSC protein, which aggregates. PRPSC is folded abnormally, and the resulting conformation, shape, is directly responsible for the lesions seen in the brains of infected cattle. Thus, although not without some detractors among scientists, the prion seems likely to be an entirely new form of infectious agent, the first one found whose transmission is not reliant upon genes made of DNA or RNA. Figure 21.17 Mad cow disease in humans. A endogenous normal prion protein, PRPC, is converted into the disease-causing form, PRPSC, when it encounters this variant form of the protein. PRPSC may arise spontaneously in brain tissue, especially if a mutant form of the protein is present, or it may occur via the spread of misfolded prions consumed in food into brain tissue. B. This prion-infected brain tissue, visualized using light microscopy, shows the vacuoles that give it a spongy texture, typical of transmissible spongiform encephalopathies. Credit B. Modification of work by Dr. Al Jenny, USDA APHIS. Scale bar data from Matt Russell. Viroids are plant pathogens. Small, single-stranded, circular RNA particles that are much simpler than a virus. They do not have a capsid or outer envelope, but like viruses can reproduce only within a host cell. Viroids do not, however, manufacture any proteins, and they only produce a single, specific RNA molecule. Human diseases caused by viroids have yet to be identified. Viroids are known to infect plants, figure 21.18, and are responsible for crop failures and the loss of millions of dollars in agricultural revenue each year. Some of the plants they infect include potatoes, cucumbers, tomatoes, chrysanthemums, avocados, and coconut palms. 21.4. Other acellular entities. Prions and viroids 527 figure 21.18. These potatoes have been infected by the potato spindle tuber viroid, PSTV, which is typically spread when infected knives are used to cut healthy potatoes, which are then planted. Credit: Pamela Roberts, University of Florida Institute of Food and Agricultural Sciences, USDA-RS, Career Connection. Virologist. Virology is the study of viruses, and a virologist is an individual trained in this discipline. Training in virology can lead to many different career paths. Virologists are actively involved in academic research and teaching in colleges and medical schools. Some virologists treat patients or are involved in the generation and production of vaccines. They might participate in epidemiologic studies, figure 21.19, or become science writers, to name just a few possible careers. Figure 21.19. This virologist is engaged in fieldwork, sampling eggs from this nest for avian influenza. Credit: Don Becker, USGS Aeros, U.S. Fish and Wildlife Service. If you think you may be interested in a career in virology, find a mentor in the field. Many large medical centers have departments of virology, and smaller hospitals usually have virology labs within their microbiology departments. Volunteer in a 528 Chapter 21, Viruses access for free at OpenStax.org. Virology lab for a semester or work in one over the summer. Discussing the profession and getting a first-hand look at the work will help you decide whether a career in virology is right for you. The American Society of Virology's website, http://openstax.org/l/asv, is a good resource for information regarding training and careers in virology.